Thank you everyone for joining us today as we discuss this pandemic that has transformed all of our lives. I imagine, like me, you're following the news of the spread of the disease, the terrible death toll, the peaks, the politics, the impact on the economy, and everything that has been really transformed by COVID-19. But what we don't hear enough about is the incredible work being done by scientists across the world to find new solutions to diagnosing, treating, and ultimately preventing this disease. This is our daily life at NYSCF and laboratories all over the world. And we wanted to bring together some of the members of the NYSCF Global Innovator community who are working on different aspects of the disease and highlight these COVID-19 research programs. They're really inspiring. I have been so inspired to see how quickly leaders in our community have responded. They've really been galvanized and are collaborating to stop this terrible virus. The latest numbers that I've seen are 78 vaccine candidates under development today, with 5 already in or preparing for clinical trials. There are over 300 clinical trials now underway, testing therapeutics with some critical readouts expected in the coming days and weeks. At NYSCF, our mission is to use stem cell research to accelerate cures for the major diseases of our time. COVID-19 is undeniably and unfortunately in that category, and really is exacerbated by the underlying conditions that is the usual focus of our work at NYSCF. Because stem cells allow you to make human models of human disease, and we work really hard to leverage this power to enable the entire disease research community to better understand and treat a wide variety of diseases. There are so many critical challenges, but I'm really optimistic that with the world's brightest minds working on all of this, we're soon going to have the real solutions we so urgently need. Just yesterday, we heard the wonderful news that our partners at Bloomberg Philanthropies and Johns Hopkins are working with New York State on the challenges of tracing and surveillance and testing. If you are interested in becoming a tracer, and we need huge numbers of people to do this, you can email contacttracing@bloomberg.org, and rest assured someone will be in touch with you to recruit you for this. It is now my pleasure to thank our colleagues at the Stavros Niarchos Foundation who support our education and outreach efforts like today's webinar. While the pandemic has changed the nature of how we convene, we're very happy for the silver lining opportunity to bring our many educational opportunities for students, scientists, and the general public online. We're very grateful for the SNF partnership as we share trustworthy knowledge and hope about the exciting scientific progress at hand, and also realistically the challenges that we continue to face. So let's get started. Feng, we hear from experts that we are not testing enough. We know that as a community, and that test results take far too long to arrive to be actionable. How do we accelerate diagnosis, scale up testing, and what are the approaches that you're using to do so?
Susan, thank you so much for inviting me to be a part of this panel, and it's really great to be here with Deepta, Shrabing, and also Scott. As you all read in the news, testing is a really important aspect of our ability to both understand the pandemic, but also as we reopen the economy, we need to have a better understanding to be able to trace, isolate, and also, test, trace, and also isolate. And the most widespread use the most widespread test that people use right now is qPCR based. And this is a test that many testing labs and also hospitals are using to diagnose patients. This is not the only test that can be used for testing for COVID-19. In fact, the COVID-19 is caused by a virus called SARS-CoV-2, and it has an RNA signature. And there are many different ways that one may be able to detect the signature. And so scientists from around the world have been developing different techniques including scientists in for-profit commercial companies. You probably have read about machines like ID Now that can help you get results within about 20 minutes. And also there are scientists in nonprofit academic areas that are working on different strategies that may allow us to scale up the testing, including using deep or next generation DNA sequencing. So my laboratory, yep.
Go ahead.
So my laboratory at MIT has been working on a couple of different ways to be able to test. One of them is based on a technique that we call Sherlock that we developed a couple of years ago that allow us to use a combination of amplification to make many more copies of that virus signature combined with a CRISPR protein that we can use to then read out the test result. This reaction can be run on a lateral flow paper strip. This is very similar to what a pregnancy test strip state looks like. And it can give users the ability to be able to get a visual readout within a matter of an hour. And back in February, we released a protocol for using this technique for SARS-CoV-2 and have sent it out to a number of researchers around the world to validate this on clinical samples. And many researchers have tested it and found that it can detect SARS-CoV-2. And then most recently, our collaborators in Thailand are beginning to scale this up and also to deploy in hospitals in Thailand for much larger scale testing. A second approach is using DNA sequencing, and my postdoc, Jonathan Schmidberg, came up with an idea called LAMP-seq, which allows patients to collect their own samples at home using an amplification technique called LAMP to be able to make copies of that and then send it into a centralized facility so that using deep sequencing, we can read out many, many samples in parallel. And this has the potential to significantly reduce the cost. And so both of these tests, they work in a complementary fashion. The Sherlock test can give you a readout within an hour or so and has the potential to be used as a point of care test or even potentially in people's homes when we can design a nice device that can simplify how this test works. And then the deep sequencing method allows large scale population level surveillance and also tracking of new infection hotspots. And these are only two examples of many of the tests that scientists are working on. And I think as we read that there is limited testing capacity, but I'm fairly hopeful that by combining all these different methodologies together, they will add up to have enough capacity to help us successfully and safely reopen our economy.
So Feng, since you joined the NYSCF community in 2016 as a NYSCF Robertson investigator, and you're a pioneer in the gene editing field. So I know one of the really critical issues is how quickly is the litmus test approach, which would be incredible, home testing, which would be really extraordinary. I mean, having a test within an hour is absolutely critical if you think about the amount of testing that we need to do to get people back to work. I mean, every hospital, every essential worker, all of the large businesses, it's really critical. So what are we looking at realistically, do you think?
Yeah, we're working very hard to try to get there, but there are some challenges. And one of the biggest challenge has been to be able to access samples and also work with clinical labs to be able to validate and also evaluate how these new diagnostics technologies compare with the gold standard qPCR-based method. For example, there are challenges getting access to different types of patient sample. Patient samples are traditionally taken as a narrow pharyngeal swab. So you stick something deep into your nose and you take a swab. You may have read in the news that one of the reasons why tests are limited is because we don't have enough swabs. We run out in the hospitals and we cannot take the sample. And so there are researchers who have been innovating on that front. For example, researchers at Yale have just released results saying that in people's saliva, there can be similarly many viruses so that you can use saliva as an alternative sample for testing. But we need to validate these new techniques on those samples. and it has been difficult to get access to those samples so that we can very rapidly validate. We're working with trying to find people to work with us. But if anyone in the audience is interested in working together to advance some of these other non-qPCR based methodologies, please feel free to get in touch. With that being said, we're hoping that within the next few weeks we can get more validation data, and then at the same time working with clinical collaborators to be able to advance this into something where people can use either in point-of-care settings or eventually in people's homes.
So helping with access, I think, is right up NYSCF's alley. So that's something that we'd be delighted to help you with. How long does it actually take, not in a queue, but to actually do a qPCR test now?
The qPCR test, yeah, the qPCR starts with taking a swab, and then the traditional protocol takes the swab and then uses a procedure to extract the RNA from the sample. And that can take somewhere between maybe 15 to 20 minutes or maybe even 30 minutes. And then that sample is put into a PCR reaction and is run for probably on the order of an hour to get the result. And then that's just the wet lab component of it. They're also registering the data, keeping track of different samples. So the whole logistics adds on top of that. I don't know exactly what the processing time is in the hospital, and it probably varies from lab to lab.
Right, but the actual work is probably like on the order of an hour and a half.
Probably a couple hours.
A couple of hours.
Yeah. To
actually do this, but I think what your point about the swabs, which I've had, they're not comfortable, but the fact that you can't get the swabs, so it's that, and then do you still run into? We've been hearing a lot about reagent problems. I know that Governor Cuomo has, and actually the Michigan governor was making a point today, and Tony Fauci actually just a few hours ago was talking about the problems of obtaining the basic materials that are required to actually do this work, which is madness.
Yeah, and these are all the challenges that we have to solve. These have to do with supply chain issues, the different reagents that we need. And so I think some of you may have read in the news a few weeks ago that the kits, the molecular biology tools for extracting RNA is in limited supply. And that has been one of the other limiting factors for testing. And many scientists from around the country, especially from basic science labs, donated their kits to hospitals so that they could use those kits for testing. And that just underscores that we have a supply chain issue that we need to solve. But there are also innovative solutions that can allow us to overcome these challenges. For example, we found that you can use a lysis solution, mix it with the swab material so that you don't have to run through a column-based nucleic acid purification kit. And that simplifies the amount of material and reduce the time that's needed for running these tests. And so I think there are different ways, but all these innovations do need to be validated and compared on a large number of samples to make sure that it doesn't reduce the standard of the test result. And so that takes time to integrate.
Well, we've seen that with the unfortunate time that was lost with the CDC test, which was really unfortunate.
Yeah.
So Deepta, Dr. Deepta Bhattacharya, a 2012 NYSCF Robertson investigator from the University of Arizona. So after we deal with having enough diagnostic tests, which Feng Zhang will solve, and we'll make sure that you have access to all of the samples that you need. The antibody testing is a critical challenge. We know that many people have likely had the disease who don't know it. There were a bunch of numbers that were published today. The very preliminary studies that by Governor Cuomo, he didn't do them, that he announced today, that it's possible that approximately 14% of New Yorkers and 21% of New York City residents have antibodies. How do we find out who is immune? Why do we care? Does it really, if people have had this, are they already immune, and for what period of time? And how do these help us develop treatments and ultimately a vaccine?
Yeah, so there's a lot of points to hit there. First of all, let me thank you. It's always good to be back in this community, and I'm glad to be part of this educational session. So thanks for the invitation, Susan, and to all the NYSCF staff. So first, let me back up just a little bit and go into the specifics of the type of test that you just talked about here for those of you in the audience who may not fully understand what it is. So this is a slightly different, a fully different type of test than what Fang was talking about. Whereas the tests that Fang was primarily talking about are diagnostics to determine whether the virus is present. The tests that you just raised, Susan, are tests to determine whether or not an immune response has been mounted against the virus. So technically the way that works is we have materials in which we stick some part of the viral protein. We do it on plastic, and then we basically take blood samples from people who are being tested and then quantify the antibodies that are in that blood sample that stick to that viral protein. And so the thinking is that this test will inform people who have been exposed to the virus at some point in the past. Once you've been infected, usually within a few days to certainly within a week, your immune response will start to generate antibodies against that virus. And this test will essentially provide some markers of that. The nice thing about this particular test is that, it basically is addressing a different question. It's not so much good for determining whether or not you have actively replicating virus, but rather you've been exposed to it at some point in the past. So these antibodies are essentially markers of recognizing that something went wrong. So the way that we are essentially testing it in the state of Arizona is sort of widespread blood testing of right now, primarily healthcare workers, frontline responders, and a handful of students on our campus. And the thinking and hope, there's two things that are motivating these tests. First of all, as a mechanism to determine how widespread the virus is within our community, and the antibodies serve as a marker for people who have been exposed to it. The second thing that is really pertinent, I think, to trying to reopen the economy is trying to get a measure as to who may or may not be already immune to the virus and are therefore likely at far reduced risk of getting reinfected, and then using that as a rational way to try and reopen the economy. So those are the those are the rationales of the test. That has lagged a little bit of the diagnostic tests that Feng was talking about, the molecular tests to look for the virus. But I think it's actually appropriate to start thinking about it now. Some of our communities are now hopefully getting over that first hump, and we can find some way to rationally instruct how to get people back to work.
We've heard a lot about the lack of veracity of the antibody tests so far. There are a number of commercial ones on the market, and we're told they, you can have them if you want it, but it's about 60%, accuracy, which is really not adequate. So how do we actually find out first, what the accuracy is, and then how do we know how long this immunity will last? Do we have any basis of that, compared to what we've seen with previous coronavirus outbreaks?
Yeah, and so let me talk about the different types of antibody tests and talk a little bit about what is the underlying problem of these accuracy issues that you just raised. So there are essentially two different kinds of tests for the antibodies. There's what are called point of care tests, and I think Feng raised that earlier, which are usually just little strips with a lancet, and people can just get a finger prick at their doctor's office, and then you get the results within 10-15 minutes or so. And it's basically like Feng said, a pregnancy test. It's a yes or no kind of answer. And I think in many ways, the pros of that are really substantial. And I think that's one of the reasons why people have been so anxious to do it. I mean, just the logistics of rolling something like that out, getting a lot of people tested, are much simpler than the test that actually we have decided to go with here in Eliza, the test that my lab has developed. So one of the problems with these point of care tests is actually as much regulatory as it is scientific. And as you mentioned before, the FDA and the CDC were a little slow on the draw in allowing molecular tests to determine whether or not the virus was present in those early days. And so they learned what they thought was a lesson from that in really accelerating the authorization of alternate tests that are being performed by academic and private diagnostic labs. And so they applied those same standards now to these antibody tests. And in many ways, it makes some sense because the actual performance of these tests isn't very difficult to do in a local doctor's office. But the challenge is that the reagents and materials that go into these tests matter a lot. And there's really not very good standardization of those materials that are going into these tests. So it's very much a garbage in, garbage out kind of assay. And because the FDA really is under the gun and doesn't have the time to really validate all of these, especially point of care tests, many of them have performed quite poorly. So for those reasons, we have decided to go with something that is not quite as easily deployed out into rural communities, for instance, but in our hands is much more reliable of an assay. So that's what's called an Eliza assay. And essentially what it requires are blood draws. So it slows things down a little bit. And then that's processed by our central lab here at the University of Arizona. We've done a gazillion quality control tests to make sure that blood samples from before the outbreak don't react at all. We've made sure that people who are absolutely confirmed COVID-19 positives are all lighting up. And so we're very happy with the performance of our assay. And we're reasonably confident that we're going to be able to provide probably better information than some of these point of care tests have been coming out with in California, for instance. Now, you asked for how long does immunity last, and unfortunately, that's hard to tell, and largely because this outbreak just started a few months ago. So I can only extrapolate from other infections, coronaviruses, and other RNA viruses, and usually the duration of immunity is quite robust. I mean, we're usually years. So I would say that the bigger challenge for how long a population will be immune for is not so much a person's own immune response, but whether the virus mutates and changes over time and then manages to evade those antibody responses.
Yeah, that's, and even if we do have the vaccine and it has to be repeated with boosters, that certainly is better than the situation we're in. So now I want to move to you, Shui-Bing. Shui-Bing, you've been a member of the NYSCF Robertson, of the NYSCF Innovator community since 2010, when you joined as a NYSCF Robertson investigator. You're an expert in drug discovery, and we've worked closely with you on the Zika virus, as well as the current work on type 1 and type 2 diabetes with Dr. Francis Collins and Mike Erdos of the NIH. And I know in January, you began to create stem cell models of SARS-CoV-2, the virus that causes COVID-19, in lung and colon organoids, which are really important for understanding the kind of havoc that this infection wreaks on the body, and for testing drugs that can stop this. This is a new and complicated disease, and there's so much that we don't understand about it. I know that that's something that you have been talking a lot about with Scott Noggle. I will just mention Scott. Dr. Scott Noggle joined us in 2008 to head up research at NYSCF. He's our Senior Vice President of Research and a developmental biologist who has also been a pioneer of our unique technology and biobank, and has been working to oversee our efforts to convert stem cells into the kinds of lung and other cells, brain cells, that are affected by COVID-19, and is collaborating with you, Shui-Bing, and top virologists and chemists worldwide, trying to leverage our biobank and our technology. But there's so much that I'm going to ask this question to both of you. This is such a new and complicated disease. I think it was today, there was a paper that came out that said that it's possible that the disease mutated differently in California and New York, creating a much more virulent strain in New York. So how can we use stem cells to accelerate research, treatment, and cures? And can you talk about the kind of drugs currently being developed? I mean, is there anything promising on the horizon? Shui Bing, do you want to start that off?
I will start.
It's
Thank
an
you.
easy question.
Yeah. First, thank you, Susan. It's really nice to come back to JAX-NYSCF and working with Feng and Deepak and Scott on this panel. So maybe I will answer your first question is how stem cell can contribute. So when we work on a new disease, usually we need to have a good model. And then for example, right now there are, as you mentioned, more than 300 clinical trials evaluating different drugs like antiviral drugs, cytokine storm drugs, and cardioprotective drugs. But most of these drugs are based on either experts' opinions or very small clinical studies. So until now, there is really no approved, FDA-approved treatment for COVID-19. So as a scientific community, we really need a very robust model to systematically evaluate this kind of different treatment and to understand whether they work, how they work, and then if it does work, how can we improve it? So currently there are three kind of systems that to use to model infectious disease, particularly for COVID-19. And first one is of course clinical samples, but this is very important, but it's very limited resource and highly vary from patient to patient. And we use animal model a lot, but unfortunately the ACE2, which is a putative receptor for SARS-CoV-2, the structure is quite different between mouse and human. So most of the mouse model, they have to use a transgenic mouse model. They overexpress human ACE2 in the mouse. Or there are some other models such as feral, like hamster that are currently developed. So it doesn't really fully recapture what happened in human. For the drug discovery, we start from in vitro model, and a lot of people use viral cells overexpressing human ACE2 receptor. Viral cell is a monkey kidney cell line, so which kind of only partially recapture what happened. Also, some study use lung or colon cancer cell line. We did a very systematic study to do the RNA-seq to compare different like current model system, and we find that neither viral cells or lung or cancer cell line recapture what happened in the patient samples. So that's how we decide to work on the human stem cells, particularly human pluripotent stem cells. In theory, human pluripotent stem cell can contribute to any type of cell in the human body, and it's already protocol reported to make like lung organoids, small intestine organoid, colon organoids, and we also contribute a little bit to that part. And in last 2 months, we really assemble a big consortium of the investigators in New York area, including the investigator at Weill Cornell, Memorial Sloan Kettering, Columbia, Mount Sinai, and JAX-NYSCF. So we built a very diverse platform. We can make 10 types of cells, organoids from human pluripotent stem cells, including like lung organoids, colon, liver, pancreatic cells, endothelial cells, cardio cells, and different type of neurons. So we systematically examine which type of cells are sensitive to SARS-CoV-2 infection. And as you mentioned, as you already know, that we moved the lung organoid and colon organoid model to high throughput drug screening. And we finished our first round of screening already, and we have some very, very promising hits. And coincidentally, one of the hits compound we find is anti-cancer drug. It was launched a phase 2 clinical trial in Spain in April 15. So now we are working with clinicians in our hospital to try to launch a phase 2 clinical trial in New York.
So that's really, really exciting. I know you're an extremely determined researcher, as is everyone who is part of the JAX-NYSCF community. You know, Scott, one of the things that I know you've heard, you know, from the many collaborators that you've been working with is that this virus tends to mutate in cells as they're being handled. So actually having an automated system to to scale this is going to be pretty critical. Do you want to talk a bit about some of that and the collaborations, kind of the overall approach? Because we used to think this, a few weeks ago, we thought this was a lung disease, and now it turns out that it's a multiple organ disease.
Yeah, so I think there's a lot of different ways that we're looking at using stem cells to address some of these different questions. As Shriving said, the traditional way that virologists have studied and grown the virus is in different cell lines that are really good for propagating the virus and expanding up the different types of studies that you can do, just the scale of the work that you can do. But they don't always recapitulate the cells that are actually infected in the patients themselves. And viruses are little machines that will replicate. Their goal is to make as many of themselves as they possibly can. And if they find themselves in an environment that's not good for their replication, mutations that sort of randomly happen can cause those viruses to, if they find those mutations, allow them to find themselves in a situation where they can replicate more. That's what will happen.
They'll just move to a different neighborhood.
Right. So it's really important to be able to find cell systems in which the virus can replicate faithfully and not change over time artificially, so that the different therapeutics that are developed can affect and target the virus that actually exists in the patient population and not just some artifact from the laboratory. So one of the key aspects that we're hoping to accomplish here is use the stem cell derived cells that we know are important in the disease as sort of faithful models for replication of the virus and studying different therapeutics that block viral replication and propagation. There's this virus is turning out to be extremely complex from a clinical perspective. It affects, it's respiratory virus. It affects the lungs, the nasal passages. But we're seeing more and more that it's affecting lots of different types of tissues. So as Shriving said, the gut, there's lots of symptoms around that you might link to the virus actually infecting different cells in the gut that leads to gastrointestinal problems. There could be, we think there's also other cells that might be attacked or influenced by the virus, in different cell types. So pancreatic islet cells, and in the case of people with type 1 and type 2 diabetes, they might be more susceptible because the virus somehow interacts. We're still very early in trying to understand how all of this works, how the different tissues are affected. So being able to have these model systems to be able to tease this apart in the laboratory are sort of critical. These are providing critical tools for understanding that and being able to progress how we understand how the virus attacks might attack and affect different tissues. We're interested have long been interested in diseases like Alzheimer's and Parkinson's disease, and where there's some indication the virus may also impact the brain in various ways, but we're still very early in trying to understand how that works and what the impact of the virus is on the different cell types in the brain. I think there was just a study that came out this week that suggested that the oligodendrocytes, the cells that wrap and ensheath neurons in the brain, might be a target for infection as well, but very early days in trying to understand this. So, being able to have access to cells that we can't easily get from individual patients, like brain cells, the cells that you can't easily go in and biopsy, using stem cells as a tool to actually make those cells is providing us with the technology that we need to actually look and understand how the disease is progressing in different cell types. And then, as Shriving said, being able to provide the raw material for testing different therapeutics that are coming out of these different drug screens. Also the types of stem cells that we're using are capturing different people's genetics. So there's many efforts, I know large consortiums working with people here in the city in New York and around the world, trying to find people that have genetic mutations that might make them more or less susceptible to infection by the virus. So as these genetic mutations are discovered, the ability to take Feng's technology of using CRISPR editing to go in and introduce those mutations in cell lines and understand what the consequences of those mutations are in these different cell types is going to be important for understanding how the virus affects different types of cells and developing therapeutics in that direction. So there's a number of different ways that we're using stem cells as a tool for modeling different aspects of the disease.
And I know that that folks at Rockefeller, I think, have been looking at
cause people to be more susceptible, particularly in cases where you wouldn't expect them to be. So the hope is that they'll find critical pathways that that make people more or less susceptible to the consequences of the virus.
So I have a question for our panel at large. We want to have a development of a vaccine that is hopefully going to put an end to this. We need to have this critical, testing, the litmus testing. We've got to roll that out very, very quickly. We've got the economic pressure, which is extraordinary. I mean, 25 million people, on unemployment. It's just madness, and that's really before we see the devastation that is happening at the countries in the world that were struggling in the first place. And are going to be completely ravaged by this. It's actually a relatively small proportion of severe cases that have been overwhelming the medical system and carrying the highest mortality risk. So figuring out, how do we figure out, who is at risk for this? And not only what can genetics tell us about this, but how do we do this quickly? Because we've never had a situation where we literally have the whole world shut down. We have people freaking out about that and, wanting to, and taking a little bit of pseudoscience and, having like chickenpox parties. And how do we actually, from a scientific perspective, get the message across to the regulators, the supporters, the collaborators, that this is something that has to operate on an entirely different timeline. So if you could each give your thoughts on that, it would be great.
I think in fairness, things have actually moved really, at a historical pace, I would say. And when I think about it from the standpoint of therapeutics, there's sort of three waves as I see it. There's the early wave of things that could conceivably help, and these are basically small molecules that maybe Shubing will be able to find sooner rather than later, that are maybe already FDA approved or have basically been shown to be safe. And those are things that can be essentially used off-label and very quickly.
Be the best.
What's that?
That'd be the best.
Yeah, so those are the quick things, right? I think Shubing could probably weigh in a little bit in some more detail than me. I'm not sure if any of those are going to be an absolute game changer, but if it could change the curve at all, I think that would be certainly helpful. The second wave of things that I actually am the most hopeful of are the monoclonal antibody therapies that are being rolled out by some of the really innovative companies like Vir and Regeneron. And essentially what they're doing is they're taking, protective antibodies, and only the protective antibodies from people who have recovered, and are scaling those up. And I actually have quite a lot of confidence that that will work and that will come onto the market a little bit sooner than a bona fide vaccine. And so you could imagine being able to use those to treat people in ICUs who are not doing well. And maybe just as impactfully, you could imagine deploying it in very specific ways so that local outbreaks are contained. And then I think after that is when we're going to see if some of these vaccine candidates work. I think I saw in the Q&A or maybe one of the chats, someone was asking, why does it take so long for a vaccine to get out there and approved? Well, I mean, the reality is that if they show quickly that it's effective, that it's safe, it will come on it will come out sooner. But it's much more difficult to evaluate those to make sure that they're actually protected from an epidemiological standpoint. That simply takes some time. So I see it as sort of a phased way to get out the therapeutics. But I've actually been really quite happy and pleased with how quickly the scientific community has come together, dropped some of the traditional barriers between different academic labs and academia and biotech and pharmaceutical companies, and recognizing that this is a common thread, and those earlier models of competition-driven progress are pretty much out the door.
Yeah, I think that's, to me, there are a couple of, I'm an optimist, but there's a couple of silver linings here. One, I think the collaboration, which is something that NYSCF has always stood for, is really supercharged. I think we will see, hopefully, much more attention to an understanding of public health and funding for public health and preventative medicine. But I think people really have to also understand that even when you have a vaccine or you have a monoclonal antibody or you have a litmus test, you have a new therapy, it has to be scaled up and rolled out. And even a vaccine program, you can't think of that as a one and done, because it takes years. It took years, 5 years of post-eradication surveillance, and that was for smallpox, which is a heck of a lot easier because you either have the pox or you don't, than this kind of invisible disease. Feng, Scott, Shui Bing, what do you think about this? How quickly we're going, and how do we get there? Because we have to get out of our house. We're all going crazy.
Yeah, so maybe I agree with what Deepak say, and maybe the most accurate treatment currently is repurposing of FDA-approved drug or off-label use for FDA-approved drug. And right now, currently in hospital, for example, at New York Presbyterian at Cornell, we have around 500 COVID-19 patients, and they are all on different drugs, like standard antibiotic treatment, just to decrease the risk for infection. And then also there are several trials testing the antiviral drugs, such as you might heard that remdesivir and flavivir, and also hydroxychloroquine, and they target different stage of the virus. For hydroxychloroquine may block viral entry, and the other drugs may affect viral replication. And also there are some other drugs, for example, to treat the cytokine storm, which happens a lot in the patient who have severe case. And there are some anti-IL-6 antibodies are currently tested to see whether it can protect patients. And more than 20% of patients, they have heart problem. They actually, particularly patient pass away because of heart attack. So in that scenario, cardioprotective drugs are also being used on the guideline to treat the patients. So,
and blood thinners increasingly, apparently too, right? Because there's so much clotting.
Yes.
Even in healthy vegan marathon runners.
That's actually the severe case. That's the problem, because traditionally people think only the people who have some obese or diabetes, they might have high risk. And turn out some severe patients, they used to be very, very healthy. And then people still don't understand why. So, So in that scenario, there's a lot of treatment options, but to my opinion, we really need a serious, a double-blind clinical trial to understand exactly which drug works and how it works. So I think that's the most important thing for the drug treatment. And I agree with Deepak that there is a phase dependency. So after 1 or 2 months, maybe we stepwise move to the antibody treatment, and after 1 year later, the vaccine will be ready. Then that will be the ideal case for prevention.
But meanwhile, Feng, you're going to be rolling out. I want this litmus test. I don't want to be waiting 12 hours after this wretched nasal swab.
Yeah, me too. I am also very optimistic about the situation. I think this pandemic is very devastating, but it's also been probably the most collaborative period of science that I've experienced. In the past 4 weeks, I've probably met more people than I would meet in a year over Zoom. Everyone who I wanted to reach and collaborate or have a conversation with, they have always been receptive to having a conversation everywhere around the world. And I think that speaks to how united and how together we are that we want to solve this pandemic for everybody who is involved. And on the sort of how do we get out of this pandemic, how do we reopen the society, I think all the speakers have mentioned really good ideas about how different stages of things can improve our ability to combat this virus. But in addition to the pharmaceutical interventions, also tests, I think technology also provides a solution. So about 6 weeks ago, me and my friend Ben Silberman, who is the founder of Pinterest, we started a nonprofit organization called How We Feel. And through How We Feel, we launched 2 weeks ago a mobile application for iOS and for Android that allows people, users, to input symptom information completely de-identified. So we're not collecting personal information like name, email addresses, or phone numbers, but just symptoms and where they are zip code wise, so that we can get a sense of where are there new outbreaks happening in the country. So far, there's a lot of interesting data that we're having able to see. We have more than 400,000 users now, and just a couple of days ago, we launched a partnership with the state of Connecticut. So we have about 2% of the state population reporting. And if you look at all the symptoms, you can see that the loss of smell and taste is the single most strongly correlated symptom for COVID-19. It's not correlated with the flu. And so I think as we collect more data, even without doing testing, we'll likely be able to generate a reasonably predictive model so that we can triage patients based on just the symptoms that they're reporting. And so I think through this combination of technology, both information technology and biology, and also our collective will to help each other to combat this pandemic, I think we'll get out of this. And we just have to stay strong and stay together.
And I think also perhaps the lack of travel has actually been helpful in a way, because you have you're not wasting so much time in airports and all of the stress of travel. So there's a little more time to think, even if we are all communicating this way. And so Scott, what do you think of what is your view of how quickly we're going to and in what order we're going to be able to make progress?
I think what I've been impressed with is how quickly people got started. I think there were a few people around the world that really sort of clued into this as a problem very, very early. And so I think a lot of people have gotten a good head start on developing therapeutics, finding drugs. So collaboration following with a group at the Sanford Burnham that did a drug screen very early on in early January using the compound library called the Reframe library. This is I think others have mentioned this, but this is a set of compounds that have already gone through safety trials, and I think it's about 13,000 different compounds that that could be repurposed and used in clinical trials. And I think one of the things that is coming out of this is that it may be possible to use combinations of different drugs to more quickly halt the replication of the virus. So the wide variety of different approaches that have gotten started very, very quickly and collaboratively around the world has been really impressive. I mean, just watching all of this take off has been very encouraging. So I have a lot of hope that we're going to make progress and continue to make progress very well over the next coming weeks and months.
Well, I agree. And I do think that although it's incredibly frustrating, I do hope that everyone around the world will stay the course and stay home, stay safe, and really just allow a little bit of the science to progress. We don't have to wait that long before we can have a much, much safer reentry. So we're now going to audience questions that are going to be asked by Dr. Liz Schwartzbach and David McCune from NYSCF.
Thank you so much. So the first question that we have is about the systemic or indirect effects. So a lot of people talk about how patients with COVID are actually dying or succumbing to the cytokine storm. So how do you look at that versus the direct effects of the actual viral entry and replication and the causes that that has on the host or us? Shubhang, maybe on the screening that you're doing, is that on antivirals or on controlling the immune system?
So we are kind of doing two-phase screening right now. In the first phase, we start from something relatively straightforward. So it's mainly focused on lung, colon, that's two major organs targeted by SARS-CoV-2. And then that's focused on the antiviral, basically blocking the virus entry, the screening we perform. And now we're kind of moving to the second phase. We try to add the immunocomponents to our screening system. And then we try to find the small molecule that can potentially block the cytokine, the recruitment of the immunocells, as well as try to find the small molecule that can block the damage of the targeting cells, such as cardiomyocyte or lung cells from the immune system. Yeah, and apparently we might need different drugs, and as Scott said, we might actually eventually need a combination or cocktail of drugs to treat the patient.
So we have a question from Reshma. So do we know if it matters how badly you have the virus, if you have a bad case or not? Do we know what the impact the viral load has on this?
I mean, right now it's pretty preliminary literature that's out there, but it certainly does seem that the higher initial inoculum, the longer the disease progresses and the worse the outcome. And I think, when we're hearing about some of the younger people that are succumbing to this disease, a lot of them are healthcare workers. And I think a lot of it almost certainly has to do with the amount of the virus that they're actually being inoculated with. What I will say, though, is that, again, we're in such early days. And I think, unfortunately, I know we need to have this information in real time, but we're not going to have any certainty, or at least slightly less uncertainty, until, a few months from now is my guess.
And Deepta, on that with the antibody testing, if you have a more severe disease, will you have a higher level of antibodies? Or if you have less severe disease, could that not be detected in an antibody test? Or is there correlation there as well?
We have seen zero correlation with that. And so basically we see antibody titers that are all, within a certain range. And in our hands, if you're positive for the antibody, then you make potently neutralizing antibodies. So it basically prevents the virus from reinfecting the cells. So again, early days, but I'm reasonably confident that once you've cleared the virus, unless you have some sort of underlying condition, that you're going to be immune for at least some period of time.
All right, and then we have a question about genetic analysis and gene editing. And Feng, I guess maybe you could talk about or kind of answer this question about if gene editing of the virus in combination with stem cell models is a possible way to study the disease, and thoughts on that. And then I know, Scott, we're doing some looking at gene editing as well, iPSC cells, so he can mention it after you if you want to take that.
Sure. There are a number of groups that are using gene editing techniques to study the virus. One of the ways is they use CRISPR libraries to be able to modify, either turn down or turn up different genes in the cell and see how that change in the cell reacts with the virus. So for example, if you want to figure out what protein on the cell surface the virus uses to enter the cell, you can knock down every single gene and see which ones prevent the virus from getting in. Or you can turn on every single gene and see which one allowed the virus to enter when it couldn't enter before. There are also researchers who are working on using the RNA targeting CRISPR systems like Cas13 to degrade and destroy the virus RNA genome from a sort of therapeutic approach, as a therapeutics approach. And these are just some examples, but there are also a lot of other ways that researchers might be able to use it to generate a better cellular, organoid, or animal models, starting from stem cells that you can differentiate to better study how the virus works mechanistically.
We have a series of questions about the different treatments that are available, whether it's using plasma to treat COVID, or the interferon B1A that helped with SARS-1 and MERS. Could you talk about, any of you, the different treatments that are currently being tested either in clinical trials or compassionate use across the different hospital systems?
So the plasma therapy, I mean, I'll briefly touch on that. Again, it's early days, and all the data that I've seen so far in sort of small trials and preprint servers, but it looks reasonably promising. And so essentially what this is taking serum or plasma from convalescent donors and then transferring it to people who are having a hard time controlling the virus. And it's very small numbers, but that looks promising so far. There have also been some studies in non-human primates that also look promising. One point of caution in data that I don't still fully understand is that some of those things have been tried for the first SARS-CoV-1, and the outcomes were a little more variable, at least in the non-human primate model. And I don't fully understand what the difference is, but the data that I've seen so far on that look reasonable.
Yeah.
Go ahead, Shubing.
No, go ahead. You continue. Yeah.
Were you going to answer interferon?
Well, you can mention the interferon. You know much more immunology than me, right?
No, go for it.
Well, I just want to say some chemicals. Basically, actually, a lot of chemical drugs that we are repurposing for COVID-19 is we learn from SARS and MERS. Yeah.
I mean, interferon, that has a lot of promise, and obviously has a lot of potent antiviral activities. And in some cases, it's actually been used in cancer therapies as well, type 1 interferons have, and it's potent. It's hard to control though, because it's a pleiotropic cytokine. I know it had some efficacy in the first SARS. I would be cautious, mainly because it certainly can trigger things like autoimmunity. I would say that if I were in the ICU, I'm not sure that would be the first thing I would want given to me.
Well, on that note, I want to, I think we're going to wrap up. I know there are a lot more questions. I know we could really go on for a while. Hopefully we'll be able to, Liz and David will be able to respond to a lot of the questions. But I want to thank all of you for taking the time to talk to us about this really critical topic, and talk about your work and educate everyone on all of the amazing optimism that we have, the incredible work that's being done by everyone. Everyone who joined today's panel is going to receive an email by tomorrow with more information about the efforts that we talked about today, as well as those from several other members of the NYSCF community. This work is so important, and I have to say it's going to be a difficult time for nonprofits over the next 18 months. So at NYSCF, we would really appreciate any of you in the audience who have been listening and are motivated to provide any support as we navigate the challenges of tackling COVID-19, in addition to all of our ongoing work. COVID-19 is something that none of us expected, I think, to have as a budget line item, and as well as the diseases that we are all working on that may now present as underlying conditions. So thank you, everyone. Have a wonderful night. Stay safe, stay indoors, wear your masks, and hopefully we will be out of this mess soon. Thank you very much.
Thanks for the invitation.
Okay, thank you, Susan.
Thank you.
Thank you.
Thank you.